I'm going to be talking about the process of RNA splicing, or the modification of precursor mRNA into mRNA that's used in protein synthesis. First, I'm going to be talking about transcription. Transcription begins with the promoter, which is a sequence on the DNA where RNA polymerase can bind to with the help of transcription factors. The promoter has several functions. It contains a starting point and also contains the tau box, which is what transcription factors recognize when binding RNA polymerase. RNA polymerase and transcription factors together form what is known as the transcription initiation complex. When that complex is formed, the process of transcription can begin, and that produces a strand of pre-mRNA. But each strand of pre-mRNA must be modified, and that's the whole idea of RNA splicing. So what happens is on the 5' end of the pre-mRNA, triphosphate guanine cap is added, or the 5' cap, and this prevents the mRNA from being degraded by enzymes in the cytoplasm, but it also acts as a signaling mechanism for ribosomes to attach. And on the 3' end, a poly A tail is added, and this basically has the same function as the 5' cap, except that it helps export the mRNA from the nucleus. Each pre-mRNA strand it contains introns and exons. Introns are the segments that are non-coding, and exons are the coding segments that are actually expressed. So what happens in splicing? Pre-mRNA combines with small ribonuclear proteins called sRNA or SNRPs, along with other proteins to form a spliceosome. The snRNA will identify the splice sites on the pre-mRNA, and the spliceosome will recognize the splice sites and bind to them. It will then cut the RNA, releasing an intron and splicing together the remaining exon pieces. This produces one continuous strand of mRNA. Once this is produced, the spliceosome will separate into its original components that can be used again in forming another spliceosome, and leaving one continuous strand of mRNA containing only exons. Okay, so I'm going to be talking about prokaryotic gene expression and the different types of operons. So first of all, what is an operon? An operon is a segment of DNA containing genes that are to be transcribed. Operons include a promoter region, operator, a repressor, and the genes. So there are two types of operons, repressible and inducible. Repressible operons are usually on, which means that transcription is taking place, but they can be turned off. An example of that is the trip operon, which I'll get to later. And an inducible operon is operon that is usually off, so there's no transcription, but it can be induced. Example of that is this lac operon. So what happens in lac operon is that a regulatory sequence on a different part of the DNA codes for an active repressor. This active repressor protein will bind to the operator, preventing RNA polymerase from binding and transcribing. So in the presence of lactose, when there are a lot of lactose molecules, eventually one of the lactose molecules are going to bind to the active repressor, causing a change in conformation and making this active repressor inactive because it can no longer bind to the operator. So that becomes an inactive repressor, allowing RNA polymerase to bind to the operator and transcribe mRNA. This lac operon is usually found in E. coli bacteria, which uses the enzymes produced by this pathway to metabolize lactose. As an example of a repressible operon, transcription is usually active, and it produces enzymes necessary for tryptophan synthesis. Similar to the lac operon, there is a regulatory sequence that produces a repressor, except this time the repressor is inactive, so it cannot bind to the operator, and that's why RNA polymerase can transcribe. But when there are a lot of tryptophan in the environment, there are a lot of molecules of tryptophan. Eventually, one of the tryptophan will bind to the inactive repressor, causing a conformation change, changing that inactive repressor into an active repressor. That active repressor will then bind to the operator, preventing RNA polymerase from transcribing. So essentially, as the cell needs tryptophan, it can produce it, but if there's too much, it will inhibit the production. It's the CRP operon. So on the lac operon, RNA polymerase needs the help of a protein called cyclic AMP receptor protein, or CRP, to bind to the operator in order to stimulate transcription. But CRP can only bind to the operator if it is activated, and it's only activated by cyclic AMP. Cyclic AMP, the concentration of cyclic AMP increases as glucose decreases, and it decreases as glucose increases. So assuming lactose is present and the normal repressor is inactivated, cyclic AMP will bind to the inactive CRP protein, causing a change in conformation to make it active, and then the active CRP protein can then bind upstream to the RNA polymerase, stimulating transcription. So assuming lactose is present and the normal repressor is inactivated, low glucose will result in there being a lot of cyclic AMP that can bind to inactive CRP, making it active and allowing it to bind to the operator, so that RNA polymerase can transcribe, producing large amounts of mRNA.